What if I told you that your liver has superpowers? It can literally regrow itself from scratch, like a real-life Wolverine. But here's the terrifying part. Scientists just discovered that alcohol doesn't just damage your liver. It traps your liver cells in a kind of biological prison, unable to heal themselves even after you stop drinking. Welcome back to Mind and Marvel, where we dive deep into the most mind-blowing scientific discoveries that change how we see ourselves and our world. I'm your host, and today, we're exploring a breakthrough that just hit the scientific world like a tsunami. Researchers at the University of Illinois, working with Duke University, just published a study in Nature Communications that completely rewrites what we thought we knew about alcohol and liver damage. And trust me, this isn't your typical alcohol is bad for you story. Your liver is arguably the most remarkable organ in your body. It's your personal detox center, processing everything from the medications you take to the toxins you encounter daily. But here's what makes it truly special. It's the only organ that can completely regenerate itself. Seriously, you could surgically remove 75% of your liver, and within weeks it would grow back to full size. It's like having a built-in repair system that puts any sci-fi movie to shame. But this new research reveals something absolutely shocking. Chronic alcohol use doesn't just slow down this regeneration, it completely hijacks the process, trapping liver cells in a state of limbo where they can't function properly and can't regenerate. Even worse, this happens at the molecular level, in a way that persists even after someone stops drinking entirely. To understand how alcohol creates this cellular prison, we first need to understand how liver regeneration normally works. Think of your liver cells like skilled workers in a factory. Under normal conditions, when your liver gets damaged, say from a virus, toxins, or even just normal wear and tear, something incredible happens. The mature liver cells called hepatocytes don't just sit there and take the damage. Instead, they undergo what scientists call dedifferentiation. Basically, they transform from specialized adult cells back into something more like stem cells. It's like a master carpenter temporarily becoming an apprentice again so they can learn new skills. In this stem cell-like state, they can rapidly multiply and then re-specialize into healthy, functional liver tissue. It's a brilliant biological system that's kept mammals alive for millions of years. But here's where chronic alcohol consumption throws a massive wrench in the works. The researchers found that when people consume alcohol chronically, we're talking about heavy long-term use, something catastrophic happens to this regeneration process. The liver cells start the regeneration process. They begin to dedifferentiate, moving from their mature state toward that stem cell-like state. But then, they get stuck. They become trapped in what scientists call a transitional state. They're no longer functioning as mature liver cells, but they can't complete the journey to become regenerative stem cell-like cells either. Imagine you're moving from one apartment to another. You've packed up all your stuff and left your old place, but when you get to your new apartment, the door is locked and you can't get in. You're homeless with all your belongings in boxes, unable to function in either location. That's essentially what's happening to these liver cells. The researchers analyzed liver tissue from patients with alcohol-associated liver disease and compared it to healthy liver tissue. What they found was shocking. These trapped cells were everywhere in the damaged livers, making up a significant portion of the liver tissue. And here's the kicker. These cells aren't just temporarily stuck. The researchers found evidence that this trapped state persists, even in patients who had stopped drinking. The cellular damage was so fundamental that it continued long after the alcohol was gone. Now you might be wondering, what exactly is trapping these cells? How does alcohol create this biological prison? The answer takes us into one of the most fundamental processes of life itself, something called RNA splicing. Let me break this down in simple terms. Your DNA is like a massive library containing all the instructions for building and running your body. But your DNA doesn't directly make proteins. Instead, it first gets copied into RNA, which is like a photocopy of specific pages from that library. But here's where it gets interesting. That initial RNA copy contains both useful information, called exons, and what appears to be junk information, called introns. Think of it like a rough draft of an essay that contains both the final text and crossed-out sections, notes, and random thoughts. Before the RNA can be used to make proteins, your cells have to perform RNA splicing. They cut out all the junk sections and splice together only the useful parts. It's like taking that rough draft and creating a clean final version. Now here's where the alcohol damage comes in. The researchers discovered that chronic alcohol use completely disrupts this splicing process. Instead of clean, properly edited RNA, the cells start producing mangled, incorrectly spliced versions. When you have incorrectly spliced RNA, you get incorrectly built proteins, and when your proteins don't work properly, your cells can't function. It's like trying to build a car with instruction manuals that have been randomly shuffled. You're going to end up with something that looks like a car but doesn't actually drive. The researchers identified a key culprit in this process, a protein called ESRP2. Under normal conditions, ESRP2 acts like a skilled film editor, making precise cuts in RNA to ensure perfect splicing. But chronic alcohol consumption triggers massive inflammation in the liver, and this inflammation destroys ESRP2 proteins. Without these molecular editors, the RNA splicing process goes completely haywire. The researchers found that the more alcohol damage in a liver sample, the less ESRP2 protein they could detect, and the more widespread the RNA missplicing became. It was a perfect storm of molecular destruction. But perhaps the most disturbing finding was this. Even in liver samples from patients who had been sober for months or even years, the RNA splicing defects persisted. The alcohol had caused such fundamental damage to the cellular machinery that it continued to malfunction long after the alcohol was gone. It's like alcohol doesn't just break your liver, it reprograms it to stay broken. Now you might think the story ends there. Alcohol causes inflammation, inflammation destroys ESRP2, RNA splicing fails, cells get trapped, case closed. But the researchers dug deeper and uncovered a much more complex and sinister web of destruction. Inflammation isn't just a side effect of alcohol damage, it's both the cause and the consequence of this cellular trapping. Let me explain what I mean. Under normal circumstances, inflammation is actually helpful. When your liver gets injured, your immune system sends in specialized cells to clean up the damage and promote healing. Think of inflammation like calling in a cleanup crew after a house fire. They come in, remove the debris, and help coordinate repairs. But chronic alcohol consumption creates a very different scenario. Instead of a quick cleanup crew that comes and goes, it's like having a cleanup crew that moves in permanently and starts trashing the place even more. The researchers found that alcohol-damaged livers were flooded with immune cells, neutrophils, macrophages, T cells, all producing inflammatory signals, but instead of helping repair the liver, these cells were making everything worse. Here's the vicious cycle they discovered. Alcohol triggers inflammation. Inflammation destroys ESRP2 proteins. Loss of ESRP2 causes RNA missplicing. RNA missplicing traps liver cells in that dysfunctional state, and those trapped cells produce even more inflammatory signals. It becomes a self-perpetuating cycle of destruction. The liver cells are sending out distress signals, which attracts more immune cells, which create more inflammation, which causes more RNA splicing problems, which traps more cells, which send out more distress signals. The researchers analyzed blood and tissue samples from patients with varying degrees of liver disease. What they found was remarkable. The level of inflammation directly correlated with the severity of RNA splicing defects and the number of trapped cells. But here's what's truly alarming. Even in patients who had stopped drinking, those inflammatory signals persisted. The liver was essentially stuck in a state of chronic alarm, unable to turn off the inflammatory response even when the original threat, alcohol, was gone. And this inflammation doesn't stay confined to the liver. These inflammatory signals enter the bloodstream and can affect other organs, the brain, heart, kidneys, intestines. It's like having a fire alarm that won't shut off, constantly telling your entire body that it's under attack. Now, before you spiral into despair thinking that alcohol damage is completely irreversible, let me share the most exciting part of this research: the potential for treatment. Remember how the loss of ESRP2 protein was a key driver of this cellular prison? Well, the researchers didn't just identify the problem, they started working on solutions. In laboratory experiments, when they artificially restored ESRP2 levels in alcohol-damaged liver cells, something remarkable happened. The RNA splicing started working properly again, and the trapped cells began to escape their transitional state. It wasn't immediate. Cellular repair takes time, but within days they could see the liver cells starting to either return to their mature functional state or complete their journey to become regenerative stem cell-like cells. The functional improvements were dramatic. Cells that had been metabolically confused and unable to perform their basic functions started producing the right proteins, processing toxins properly, and even beginning to divide and regenerate. But that's not the only potential treatment avenue. Remember that vicious cycle of inflammation? The researchers found that when they used anti-inflammatory treatments to break that cycle, it also helped restore liver function. By reducing inflammation, they could protect the remaining ESRP2 proteins from destruction and even encourage the production of new ones. It's like finally turning off that fire alarm and giving the repair crews a chance to actually fix things. Now, we're still in the early stages of translating this research into actual treatments for people. The experiments I just described were done in laboratory cell cultures and animal models. Human clinical trials are still needed, but the researchers have identified several potential paths forward. They could develop drugs that directly boost ESRP2 levels, or medications that protect ESRP2 from inflammatory damage. They could also explore more sophisticated anti-inflammatory approaches that specifically target the liver without suppressing the immune system everywhere else. There's even the possibility of personalized treatments. Since the researchers can now measure RNA splicing patterns in liver tissue, they could potentially tailor treatments based on the specific splicing defects in each patient. Perhaps most importantly, this research gives us hope that even severe alcohol-related liver damage might not be as permanent as we once thought. If we can break that cycle of inflammation and RNA missplicing, we might be able to unlock the liver's natural regenerative powers even after years of damage. But the implications of this research extend far beyond just alcohol-related liver disease. This study has opened up entirely new ways of thinking about chronic diseases and cellular dysfunction. RNA splicing defects have been found in many other conditions: cancer, neurodegenerative diseases like Alzheimer's, autoimmune disorders, and age-related cellular dysfunction. This research provides a roadmap for understanding how inflammation can trap cells in dysfunctional states across many different diseases. Think about it. If alcohol can trap liver cells in a transitional state through RNA splicing defects, what about other organs? What about brain cells in neurodegeneration, or heart cells after repeated injury, or lung cells in chronic inflammatory diseases? The researchers noted that some of the RNA splicing patterns they found in alcohol-damaged livers were similar to patterns seen in normal aging. This raises fascinating questions about whether some aspects of aging might involve similar cellular trapping mechanisms. From a prevention standpoint, this research emphasizes something crucial. Inflammation is far more dangerous than we previously realized. It's not just about temporary pain and swelling. Chronic inflammation can literally reprogram your cells to malfunction. This means that lifestyle factors that reduce inflammation, like regular exercise, a healthy diet rich in anti-inflammatory foods, stress management, and adequate sleep, aren't just generally good for you. They might be protecting your cells from becoming trapped in these dysfunctional states. It also raises questions about environmental factors. If alcohol can cause this type of cellular reprogramming through inflammation, what about other toxins we're exposed to? Air pollution, processed foods, chronic stress, certain medications. The research methods used in this study, combining single-cell RNA sequencing with epigenetic analysis, are cutting-edge techniques that are just becoming widely available. As these tools become more sophisticated and accessible, we can expect to see similar discoveries about other diseases and conditions. We're potentially on the verge of a new era of precision medicine, where we can identify specific molecular defects in individual patients and design treatments to correct those exact problems. Instead of just treating symptoms, we could be fixing the fundamental cellular machinery that's gone wrong. So, what does all of this mean for you personally? Whether you drink alcohol or not, there are several important takeaways from this research. First, if you do drink alcohol, this research underscores the importance of moderation. We're not talking about having a glass of wine with dinner. The severe RNA splicing defects were found in people with chronic heavy alcohol consumption. But it also suggests that the damage from heavy drinking might be more persistent than we previously thought. Even if someone stops drinking, the cellular reprogramming can continue for months or years. Second, regardless of your alcohol consumption, this research highlights the critical importance of controlling inflammation in your body. Chronic inflammation isn't just uncomfortable, it can fundamentally reprogram how your cells function. This means prioritizing anti-inflammatory lifestyle choices becomes even more important. Omega-3 rich foods like fish and flax seeds, colorful fruits and vegetables high in antioxidants, regular physical activity, stress management techniques like meditation and adequate sleep, all of these help keep inflammation in check. Third, if you have a history of heavy alcohol use, It's crucial to work with healthcare providers who understand these new findings. Traditional liver function tests might not capture the full extent of cellular dysfunction that this research has revealed. As this research progresses, we may see new diagnostic tools that can detect RNA splicing defects and cellular trapping before traditional symptoms appear. Early detection could lead to earlier intervention and better outcomes. For anyone struggling with alcohol use disorder, this research actually provides additional motivation for seeking treatment. We now know that every day of continued heavy drinking isn't just causing more liver damage, it's making the cellular reprogramming more entrenched and harder to reverse. But there's also hope in these findings. Understanding the mechanisms of how alcohol traps cells gives us targets for new treatments. Future therapies might be able to unlock these trapped cells even in people with severe liver damage. This study represents just the beginning of a new chapter in our understanding of cellular dysfunction and regenerative medicine. The researchers have opened a door to a whole new world of therapeutic possibilities. Over the next few years, we can expect to see clinical trials testing ESRP2-based therapies, more sophisticated anti-inflammatory treatments specifically designed for liver disease, and potentially even gene therapy approaches to restore proper RNA splicing. The combination of artificial intelligence and these new cellular insights could accelerate drug discovery dramatically. AI systems could potentially design molecules specifically to restore RNA splicing function or protect ESRP2 proteins from inflammatory damage. There's also the possibility of more advanced regenerative approaches. If we can understand exactly how to reprogram trapped cells back to a healthy state, we might be able to stimulate liver regeneration in ways that seemed impossible just a few years ago. On a global scale, this research could inform public health approaches to alcohol-related liver disease, which affects millions of people worldwide. Understanding the molecular mechanisms could lead to better prevention strategies and more effective treatments. We're also likely to see this research integrated with other fields: immunology, neurology, cardiology, and oncology. The principles of cellular trapping and RNA splicing dysfunction probably apply to many different diseases. Perhaps most exciting is the potential for personalized health monitoring. Imagine being able to detect the early signs of cellular dysfunction through simple blood tests, long before any symptoms appear. This could revolutionize preventive medicine. So there you have it, a groundbreaking discovery that changes everything we thought we knew about alcohol, liver damage, and cellular regeneration. Chronic alcohol use doesn't just damage your liver, it reprograms your cells to stay damaged, trapping them in a biological prison through RNA splicing defects. But within this sobering discovery lies tremendous hope. For the first time, we understand the molecular mechanisms behind this cellular dysfunction, and that understanding points the way towards new treatments that could unlock the liver's natural, regenerative powers. This research is continuing right now in laboratories around the world. Scientists are working on ways to restore ESRP2 function, break the inflammation cycle, and reprogram trapped cells back to health. If discoveries like this fascinate you as much as they fascinate me, make sure to subscribe to Mind and Marvel and hit that notification bell. We're living in an absolutely incredible time for scientific discovery, and there are new breakthroughs happening every week that could change how we understand ourselves and our health. I want to hear from you in the comments. What aspect of this research surprised you the most? Have you or someone you know dealt with alcohol-related liver problems? What questions do you have about RNA splicing or liver regeneration? Thank you for joining me on this incredible journey into the cellular world. Until next time, keep marveling at the amazing science that surrounds us every day.